Hello, everyone. Thank you for joining this installment of Voice Hobson Institute's Breaking Ground discussion series. My name is AJ Bushy. I'm in the communications department here at BTI. And for this month's Breaking Ground, we welcome back Dr. Joyce Van Eck, who was our first ever Breaking Ground guest way back in June of 2020. Thank you so much for joining us again today, Joyce.
Sure. It's hard to believe it's been 2 years. My goodness.
I know, right?
Yeah. Congratulations on the success and keeping this going.
Oh, well, thank you. Thank you. So Joyce is a plant geneticist here at BTI, whose lab is focused on biotechnological approaches to studying gene function and crop improvement. And today we'll be talking about her broader work with the plant biotechnology community to help improve plant security. And so first, couple housekeeping announcements for those of you who haven't seen a Breaking Ground before. It's about an hour long. We do a half-hour discussion with me and Joyce, and everyone else will be muted, and then you can ask questions. In this latter half hour, we'll do a public Q&A with all of your questions. And I've enabled a live transcript function, so you should be seeing closed captions. If you'd like to turn this off, you can do it with a live transcript button. Down below, you'll see a little box that says CC. So you can click hide subtitle, which I just did. And so that's it. So let's go. I'm really excited today. We'll be talking about biotechnology, food biotechnology with Joyce. And let's kick it off. Could you tell us a little bit about yourself, Joyce?
Sure. Grew up in Pennsylvania, small town called Plains, only about 2 hours from here. I got hooked on science in elementary school. Actually, I went to Catholic school, and then I was really focused on math and science. And that's where I got my love of science from. And when I was in high school, I decided I wanted to be a doctor, specifically a pediatrician, because I wanted to help people. But when I got to college, I never had to do dissections when we were in high school. We didn't do that as part of our biology classes. And then when I got into undergraduate classes, I was like, this just isn't for me. It's a necessary part of this whole doctor training. I need to find something else. And I was still interested in helping people. I was very much interested in genetics. Did some searching around, and I learned about plant breeding and genetics, which I didn't know when I was in high school. I knew about that field. And I had a love of plants from my parents. We always had a garden. And I was like, well, let me give this a try. And I never turned back. And I was on the kind of trajectory to be a maize breeder. And then I learned about plant biotechnology. And I got very much interested in plant biotechnology, and then decided to go on to graduate school, and that would be the focus of my career.
And you mentioned, how you want to help people in your research. Could you talk a bit about food security and how that ties in?
Yeah, sure. So, I think it's, I don't know if people realize that we rely on just 15 crops to provide about 90% of our food supply. And the big ones, which are rice, maize, and wheat, make up about 2/3 of that supply, and then potatoes are like the fourth one that link into this. And here's this definition on food security, which I found from the United Nations Committee on World Food Security. So we really need to be looking at that to make sure that we have the enough food to feed people. Is that going to be important, especially we hear about the growing population. There's also the amount of land that's available because of development for farming. And then, we're all very much aware of what we're seeing even now is the stresses of climate change. So we really need to, be looking at diversifying our food supply, looking at, potential other plant species that we could look at to improve. And it's amazing that there are 50,000 potential edible plant species, but again, we rely on just 15 of those for the majority of our source of calories. So I think the other thing is that with different pressures, again, from climate change, and we're seeing an increase in pests and diseases, and we not only thinking about potential new crops, but how do we give our current staple crops the resilience to continue to be productive So that's where, I see that, plant biotechnology is a tool. It's one of the options we have to help with that food security.
Yeah, that's one thing I'm not sure how many people are aware of is that, as the climate changes, pests and pathogens are expanding their reach. So you're getting, fungal pathogens in places where they weren't used to before, and then times of the year where they didn't used to be a problem. And then all of a sudden now you get rain when you didn't used to have rain. And
Mm-hmm.
you have fungal.
Yeah, exactly. And we'll talk a bit about that in the next couple of slides when we get to crops in particular, about where that's having an effect.
Sure. Let's talk about them. What
Okay.
do you have in mind?
Okay, great. So I didn't know if you wanted to switch to that slide or the next. I think that's the next slide. I think there we go. I just wanted to have those beautiful images to help illustrate what we're going to talk about. So, so because of these different stresses that are coming on plants, we're seeing some issues that we haven't seen before, and they're really being emphasized compared to what they had been in the past. So, two crops near and dear to my heart, or two foods, are chocolate and coffee. We also see a similar, we're not going to talk about that today, but with banana, these diseases that are coming in and really threatening this supply. So you can see here those pods. I think cacao is so cool. I worked with this when I was an undergrad. The pods grow off the trunks of the trees, and you'll see on the right there, that one image with those white kind of mucilaginous. Those are the inside. Those are the seeds. So they're covered by this kind of gooey substance, and inside are the beans, and those are roasted, and then they're used for making chocolate. But you can see that pod in the bottom. That's a disease that's coming into cacao, and it's really you can find articles in the popular press about how it's really affecting the good quality chocolate that we have there, and it's plant breeders. One of the things that plant breeders will do will look for sort of the relatives or the germplasm that have a natural resistance to these diseases, and it's just not there. So they've got to, really now find a way to help improve resistance to this particular disease. And it's a similar story with coffee that you'll see on the right. You'll see that the leaves are affected, but not only are the leaves affected, but the beans are affected as well. So you'll see this image on the upper image in the hand. Those are what the coffee berries look like, and inside are the seeds, and the seeds are what's roasted. But you'll see in that lower image, those are beans that are affected by the disease. So it's not just the leaves that are affected, but the beans are also. And this has been, there's one coffee in particular, which is called Arabica, which is considered like the higher quality coffee. Those are primarily grown in upper regions. But now with the temperature increasing, even those upper regions of where they're grown are getting warmer. And that's just causing this disease to happen faster. And people are scrambling, researchers, plant breeders are scrambling to find that natural resistance and start making crosses so that we can have coffee that's resistant to this pathogen.
So let's talk a little bit more about those methods, and not just you mentioned like kind of wild relatives as reserves of genes. How does plant biotechnology kind of tie into this and all the different solutions for protecting our crops?
Yeah, sure. If we could go to the next slide. I'm just going to move my image here. So these are all the tools that we can use to help improve crops, and they are indeed used. And it's my opinion that when we are dealing with these situations, that we should take advantage of every tool we can to help food security. And I've listed some of those here. So plant breeding, I looked up a definition of plant breeding, and it's described as the art and science of changing the traits of plants to produce the characteristics that are desired. And this could be done in the greenhouse or field. And as you know, plant breeding has been around for a long time, and plant breeders do an incredible job. I just don't think they get the appreciation that they deserve for what they've done for our food supply, and what has changed our crops. And I think sometimes people just aren't aware of the behind the scenes, but if they were aware, they would be more appreciative for what goes in and what plant breeders have been doing for a long, long time. The next field is genomics or genome mapping. And this is kind of like this the DNA sequence. You've heard of the human genome. We do this for plants as well. And this is a roadmap to help identify genes. Can we identify the genes that plant breeders can use or other researchers can use for looking at, say, disease resistance, insect resistance, or even improving yield? Once you have those genes and you've identified them kind of in this roadmap, you then need to determine and to verify the function of the genes. And that's something called functional genomics, to make sure that you're working with the right gene in its right form to get the outcome that you desire. Genetic engineering, this definition is similar to plant breeding, where it's the art and science of changing the traits of plants to produce desired characteristics. This is done in the lab. So it's different from plant breeding, which is done in the field, and we do this in the lab. And genetic engineering is really a very key part of the functional genomics part of this toolbox of what we can use for crop improvement. So using genetic engineering can really help us identify the function of genes for improvement in the future. And they're also used not just looking at the function of genes, but also for improvement of traits. And we'll talk a little bit about that later. The newest tool on the scene is gene editing. CRISPR, a lot of people have heard about CRISPR in relation to it, what's being done in plants in relation to human genetic diseases. And that's a tool where we can make these precise changes in the DNA sequence, really tweak it. And this is a type of tool that those of us who have spent our careers in plant biotechnology, we're just dying to have. Can we be more precise? Can we make these changes happen at the DNA sequence itself? And so basically what I kind of look at, CRISPR is, and I don't know if many people are aware of this, but there are natural mutants that can occur in a plant species. And I'll tell you, for instance, there's a tomato. If people have grown tomatoes, there are these long, lanky, uncontrollable tomatoes that grow. Cherry tomatoes are often that way. But then there are the more compact, or what are, these they're easier to grow, and they're called determinate. So those are the determinate, the more compact, and then the wild, long, crazy growing tomatoes. Well, those determinate tomatoes, especially you'll often find these are sold as being patio tomatoes. That was due to a natural mutation that happened in a field back in the late 1920s. And a grower saw this more compact tomato plant. And then over time, they identified what gene was involved, and then breeders started to use this mutant as a source for especially for commercial tomato breeders. They to make the plants more manageable and more realistic to grow in an agronomic setting. And I look at CRISPR that way. CRISPR is this way to create these precise mutations that then can help benefit different plant characteristics that people want to achieve. And it can also do it pretty rapidly. We've been doing some work in my lab to show how we could basically fast track improvement or fast track domestication of unutilized species by using CRISPR. So again, these are all the tools that are the option. Maybe there are some of these tools you don't need. You go strictly with plant breeding, or there's a combination of one or several tools. But again, I really think it's my opinion that we need to take advantage of whatever we can to make sure we have a secure food supply.
Yeah, I guess when I think of traditional plant breeding or conventional, how you want to think? I think you take the pollen from, you have a big tomato, and then you have a smaller tasty tomato, and you take the pollen from one flower, mix it with the other, and then you hope to get a big tasty tomato. But that can take years and years and years, and you might get some other things that you don't want along with that. So it takes a long time, and it's not precise. So that seems
exactly. Yeah, you have to sort through a lot of material. I mean, when we go through genetic engineering and gene editing, it's not that breeding isn't part of it or that evaluation doesn't happen. It certainly happens, but it helps bring that variability into play for plant breeders, kind of like having a palette of paint. Where you have all these different paints that you could choose from, these colors, and be able to put it together. So providing that kind of palette of different plant lines that have the variability, say for fruit size or plant growth habit, really helps the process of crop improvement.
And there's marker-assisted plant breeding, right? Is that a thing?
Mm-hmm. Right, which is in a way to be able to identify. You wouldn't have to grow a plant to full maturity to determine that, and that really helped revolutionize plant breeding and shorten the time for trait evaluation. Yep.
So yeah, no, that's big in cassava, I think, which you might, instead of waiting like 5 or 7 years to see, you can find out, pretty much right.
Right, exactly.
So we can't really talk about this without talking about, the GMOs and some of that controversy. Can we talk a little bit about, how that's gone over the past few decades and where how things are going there?
Yeah, so I mean, I really like this. I found this online. It's from ISA, which was an organization based here at Cornell originally. I'm not sure if they still are or not. But this shows that since 1996, so like almost 30 years now, where we started. It started in the U.S. and where it's grown in the countries where these improved plants are being grown. There's a little key here at the bottom that shows these little icons with soybean and maize and cotton and where they're grown and how much is being grown. And this is as of this is the latest information I can find, and this is from 2019. So I just, there's a lot of success stories here, but I just wanted to focus on 2 of them. And this is that BT eggplant. And eggplant isn't a huge, interest in food in the U.S. It's, this compared to some other food crops, but it certainly is in Asia, specifically South Asia, in their cultures. And one of the problems with growing eggplant was it is really susceptible to this insect called the eggplant borer. I think that's what. Let me take a look at my notes here. I always remember it as this acronym, but it's this eggplant. Yeah, it's this larvae that bores into the eggplant fruit shoot borer, EFSB. And it really devastates. It devastates not just the plant, but the fruit itself. And the way farmers have been dealing with this is tons of pesticides. That was the only way that these farmers in countries like Bangladesh could control these insects. So pesticides are expensive. They're not great for us, right? And I know someone who is connected with this work, and he said that some of the farmers in these countries wouldn't even let their own family members eat the eggplant that had been doused with these pesticides because they knew that this was a problem, having their families eat this. So over time, this approach was taken for BT eggplant, and BT is this Bacillus thuringiensis, also referred to as BT. It's a naturally occurring soil bacterium that produces this protein that happens to confer, this resistance. And BT, for those people who are organic farmers, you might be pretty familiar with BT because it's often used for controlling insects. So there's a group of researchers who developed this, did this genetic engineering with the BT gene and developed this insect-resistant eggplant, which has really made quite the impact in these countries, in Bangladesh and the Philippines. And again, this person who I know is involved in the work said that the farmers who were growing it, and then there were farmers who didn't know about it, and they'd see these farmers who were growing this incredible eggplant that they didn't need to use to put pesticides on, and they wanted in on the game. They wanted to find out, how they could get this eggplant. So, it's just an amazing story about, and it's not just, like we talk about coffee and chocolate, and it's not just that we're not going to have our morning coffee and we're not going to have our Hershey's chocolate. It's the lives of these farmers. It's the livelihood. It's their families. It's the economy in their countries. So it's a bigger impact than just us not having getting these items. It really has a larger effect than that. The other story that's one of my favorites is the papaya. So papaya, there's a disease called ring spot virus, papaya ring spot virus. And this also affects both the leaves and the fruit itself. and it was no known resistance, where breeders could go in and start doing crosses and come up with the resistant. No chemical control that you can use against viruses. So a group of researchers, actually here at Cornell and at universities in Hawaii, came with an approach where they did genetic engineering, and they came up with a virus-resistant papaya, and it pretty much saved, and it's not an exaggeration, saved the papaya industry to have this resistant fruit. So just to show that this isn't anything new. This has been around for a while. You see this map. It started back in 1996, and again, this wasn't just people doing genetic engineering. It involved a lot of different groups. It involved plant breeders and entomologists and pathologists, and so it's really a big diverse group that you need to be able, and the farmers to be involved in this work. But it certainly has shown benefits, especially to farmers.
So there's clearly a lot of promise and need for these technologies, but I understand there are some bottlenecks, sort of kind of in the way of it really achieving its promise.
Right. So over time, this is really, so as I mentioned before, like plant biotechnology, one of the primary uses of it is a fundamental tool is in understanding gene function. And that gene function can be key for crop improvement. And in this slide, I improved this transformation isn't a crisis. I didn't say that. That was attributed to a National Science Foundation program officer last year at a meeting of the PlantGENE research program, where they were talking about some problems, and this was his quote: "Transformation isn't a crisis." And this crisis is actually impeding plant science research worldwide, and also, it's really a big bottleneck. And part of that is the capacity to do transformation can't keep up with the demand. So we have labs who do their own transformation, but then there are also these facilities that do it. But they just can't keep up. And as I mentioned before, this is having a negative impact on plant research. And this is worldwide, not just in the US. And part of that, there's several reasons for this happening, is that the demand for transformation or plant biotech, so because there's plant biotech, I'll say transformation and genetic engineering. So I use those interchangeably, but they all pretty much mean the same thing. That demand skyrocketed with the advent of gene editing. People really saw that value of being able to tweak those genes right at the DNA sequence for fundamental basic research and also for crop improvement. So when you have all these groups who don't have the capability to do the transformation in their lab, they really are looking for people to do it. And it's just, we just can't keep up. And part of that is because it's labor intensive. That's mainly because of something called the plant tissue culture process, which is very hands-on, and part of, this really takes a lot of people power. And it's difficult to find people who are trained in this technology, both in the theory, but also in the methodology. You really need to understand what you're doing and why you're doing, not just having this hands-on, so that you can troubleshoot, that you can develop new methods. And it's difficult to find people at all levels, technicians, it's at the leadership levels all the way through. And this is not specific to academia, this is academia and industry as well. And a part of it is that we need to think about methods differently. We need to, one of these is we have these methods where we can deliver these plant gene components into plant cells. And we won't go into talking about those, but I can tell you that those are inefficient cell delivery methods. And that makes the work expand. Whereas if you had something that was more efficient, you wouldn't have to have so much input to get a good output through the pipeline, if that makes sense. So having more efficiency in the beginning. Part of this crisis is that we really need to bring people together working. We've got people who are working in different labs and in different public sector and private sector, but we really need a more cohesive effort if we're going to crack this crisis and this bottleneck. So one of the ideas that we want to get into is, thanks again for giving me the opportunity to talk about PlantGENE. So how did this PlantGENE come about? It stands for the Plant Genetic Engineering Network. Last year, we have a society, a conference called Society for In Vitro Biology, and it's animals and plants. And we had a session where we had a panel discussion. I was co-chair of that discussion called Building Partnerships to Break the Bottlenecks of Plant Biotechnology, or something like that. And in this panel discussion, we really asked the audience, where do you see the problems? What needs to be done to help the situation? And, a lot of conferences, people talk about great ideas and things happen, and people go away and nothing happens. Myself and others were determined to not let this happen, because we know how important it is. So we formed, so myself and some volunteers formed what I call a think tank, to try to take all this input we got and how can we form some sort of concerted effort to start addressing some of these issues. And we came up with this idea for the Plant Genetic Engineering Network, or PlantGENE. We submitted a proposal to the National Science Foundation to something called the Research Coordination Network Program. And we did that at the end of December last year, and found out July 1st that we were officially funded, which was very exciting for us. And so it's a group of, we'll see a picture later on of myself and others that I've known for a long time, or people who spent their careers in plant biotech. And I put this diagram together to show how this is all interconnected. So we've got PlantGENE in the middle. What's really important is that we want community input. This is a community-driven initiative, and we want to make sure that we're hearing from the community, from the users, of what are the needs and how best can PlantGENE help. And the way that PlantGENE can help is to making connections, being that kind of hub of a wheel. We want to assist with research coordination. What are the ideas for breaking these bottlenecks at the research level? How can we pull people together? How can we connect researchers? How can we find funding opportunities? I mentioned training and finding people who are trained in these areas. We really need to facilitate training, internships, and this is both industry and academic labs. We need buy-in, and we need funding. That's what makes things happen. You can have ideas, but you need people, and you need the funding to make this kind of dream come true. Virtual workshops. So we have a website, which I show over here, and all the different parts we want to have of the website. But we also want to have these virtual workshops, because we could have reach. We could have bigger reach by having virtual around the world. And if we try to do some sort of hands-on, which we do have some plans for that, but we couldn't get as big a reach with hands-on as we could with virtual. And I mentioned some parts here that these would be community members. We're going to enlist them to help. These could be crop-specific workshops. It could be on techniques. It could be the theory behind these different areas of plant biotech. We also, we have these transformation facilities where these groups will do transformations as a service, do the transformations, do the genetic engineering, and then give researchers the plants back. We have some great facilities, but they haven't really been pulled together. We need to form this network to get people talking, to find out where the needs are and how they can best address the needs of the users. A big part of this will be training, hands-on workshops. What I'd really like to have is, we have this great REU program here. I'd like to see an REU program specifically for transformation facility, hosted at transformation facilities, and have them provide the training for this, and then have students form their own network. So this is kind of the big picture. A lot to do, but I can tell you, we're super excited about having this opportunity.
Yeah, so you mentioned, I see trainings and workshops in numerous of these little, parts of the diagram. So that sounds like it's, like a major focus of PlantGENE. That sounds great.
We need to train the next generation. We need the people power to have that happen. And one of the things I wanted to say is I very much applaud the National Science Foundation, NSF, for having the vision to fund this this initiative. But we need more. This just shouldn't be left up to NSF to shoulder the responsibility. We need other agencies to help. We need foundations to help. We really want to connect with academic labs and industry to help make this training happen, because the training is going to take funding. Just to have training in people's labs, it takes supplies, but if you're asking students, you're asking people to come to your lab, say for a summer, they need some sort of source of income. They need something that we can help with these stipends or places to live. And with the funding we have from NSF, we're so much appreciative, but it's just not a big enough pocketbook to allow us to be able to do all that needs to be done. So part of what PlantGENE is doing is trying to seek funding from other places to help, and not just PlantGENE, but members of the community. That will be one initiative to make sure that we can, make sure we have the support that we need.
And are you doing this all yourself, or?
Nope, we can go to the next slide, right? I think it's the next one. Oh, no, okay. Oh, I guess I thought we had a slide with the steering committee, but I guess we
no, I don't think so.
Oh, that's okay. So what we so no, I'm not doing all myself. It's a team effort. And so we have a steering committee. There is, I'm trying to think off the top of my head, Wayne Parrott from the University of Georgia, Vina, and Nigel Taylor from the Danforth Center. We have Bill Gordon Cam from Corteva AgriScience, Kunsub Lee from Iowa State University, and Heidi Kepler from the University of Wisconsin are part of the steering committee. Oh, good, we are. Great. So so this is Bill. I mentioned Bill from Corteva AgriScience, Heidi from left to right, Wayne, Vina, myself, and then Kunsub. So this is our steering committee, and Nigel wasn't at this, but Nigel is part of the group, but he wasn't at the conference, so he's not in the picture. So this is the steering committee. I've known some of these people quite a long time. These people have spent their careers in plant biotech. But then down from that, we have our advisory committee. So that's a group of people, both international, industry. We have someone from the Corteva Human Resource department, because we really, of course, have a focus on training a diverse work group and giving people opportunities. So having her guidance will help. But then it's the community. So we're there to just help steer, but we need the community to help out and to volunteer and to step up and to be part of this as well. And we have a website, and we also have membership so people can sign up. We have a Twitter account. You put it up in the beginning at PlantGENE, and we really are, building this community. And again, I have to remember we just officially started July 1st, but we were ready to go.
Okay, and we're about ready to accept some questions. If anybody wants to throw their questions in the chat box, but I guess I'll kick one off that I just thought of is that is just, I guess, the demand. Like, do you see, when it comes to demand of different species and methods, right? Every species needs kind of its own method, of how to best transform it. Where do you see the demand right now, and is that changing from 10 years ago, and do you see it changing 10 years in the future?
Yeah, it's definitely changing. In the beginning, it was basically what worked, the methods that worked well, and I won't get into the details, worked well for what we call model species. So one of the big models, kind of the white mouse, the little mouse of human research, our model was tobacco, because it worked really well for plant transformation. And you could get your answer quickly and then think about other plant species. But over time, that grew, and I can tell you, one of the biggest demands is tomato, and that's because tomato is a great model species, because it's an important crop species, right? But it's also an excellent model species because of all the resources that are available, and a stellar genome sequence, all these mutant lines. It's got a long history, and the transformation method is well worked out. So there's quite a bit of demand for tomato, but over time, that's now grown into other crops. Maize, maize is a big one, soybean, and those aren't as easy as tomato. So that takes a lot more input than tomato does. So some of this coordinating research that I talked about, and there are groups already working on this, is how can we make those methods better? How can we make them more efficient? How can we, have them be done by not just people specialized in the field for doing this work?
Okay. I think I accidentally had the chat box setting so no one could type in it. So I think I fixed that. Hopefully I fixed that. That was I've never done that before. So if anyone out there has some questions, and I guess I do have another question about the coordinator. Are you still to hire someone?
Yeah, thanks for that free advertisement. So part of PlantGENE is that, members of the steering committee, we all have our research programs. We are excited about getting PlantGENE going, but we really need help with everything, that all the coordination that we want to do, and coming up with ideas for how we can expand the reach of PlantGENE. So we're looking for a PlantGENE project coordinator to be involved in the project. The project goes for 3 years. That was, having a full-time coordinator, and the amount of funding that we had was, the extent of what we could do with the amount of funding. But we really need somebody to help. And there's a link on the career site of BTI where you can apply, or people can apply for this position. But it would be, working with the website, getting community to, being a real connector, helping make those connections with people, get people talking, help with the virtual workshops. And some of this is described in the ad that's on the BTI website.
And we got a couple questions. One is from Juliana. Great talk. I believe food diversity could also have a positive impact on the carbon footprint in agriculture when there is an investment in domestication of local species to supply local markets. It's a good comment.
I completely agree. I mean, why, there are species where you could take and you can try to make them more drought tolerant. But there are species in their natural environments that are already drought tolerant, but they need some tweaks. They need some improvements. Like there's one plant species I'm aware of that actually there was a group in Africa who asked me about this. It's called fonio. It's great nutritional content. It is used at some level, and it's adapted to the environment, but it needs some improvement. One of those is that the seeds, they kind of call it shatter, where they fall off the plant. And so I agree with that comment by the person who put it in the chat, looking for those kind of little gems that are already adapted to the environment, have, these tolerances, but just need to be brought, kind of elevated to an agronomic level where they're realistic to grow on a larger level.
Some of that is making them easier to harvest.
Yeah, definitely. Yep, easier to harvest, increase the yield.
In designing your new PlantGENE community, have you considered using system science methods to design a new system like this? I don't know what system science is.
Yeah, no, I haven't. That's an interesting comment, and I'd like to learn more about that. But we haven't considered that.
That was from Pamela Sedelko. Thank you, Pamela.
Okay, I will take that name down.
And Brian Dowd Uribe asked, a very interesting initiative. 2 questions. Could you tell us a bit about the role of social scientists in PlantGENE? And also, how do farmers from different backgrounds and geographies plug into these efforts? That's a good question.
Yeah, that's a good question. So right now, this is kind of our platform, the original platform for PlantGENE, what we can do at this point. But I agree with the social sciences comment. We're hoping that this coordinator can help make us some connections with social scientists. And you have that little grid. I imagine my vision for the future is that grid, that schematic will grow, and we'll add in the social scientist. That's a really important part of it. Excuse me. And then as far as access for farmers to this technology, that would, or sort of the improvements. So I think that the benefit of this is kind of breaking those bottlenecks so we can get improved varieties, and that would be dealt with on a kind of a case-by-case basis, a crop-specific basis, and a regional basis. And that might be something that would have to happen beyond PlantGENE. I hope we can have like a version 2 of PlantGENE, because right now we only have 3 years. So we have to deal with the immediate problems now, the immediate ways to try to break these bottlenecks, and then hopefully we can grow the initiatives and have something in place. We talk about farmers. I think a big part of this is this community science and outreach that needs to be started to bring people along the path with us.
So there's 3 years of funding with this NSF?
3 years of funding, yeah. 3 years of funding, yeah. So then, like I said, if we have before NSF, putting this into NSF, reached out to other agencies and foundations to help, strengthen this, to broaden it, to be able to continue. I just don't want to get 3 years, we're done. That's not what I want to have happen. And I just don't want it to be even just within, we see it just not our group, but just help others to kind of thinking about this and kind of grow that tree for PlantGENE, and have others look for funding to be able to continue this as well.
Sounds like a good plan. And I just put the website for PlantGENE in the chat box if anybody wants it. Here's one from David Silverman. Hi, David. Can microarray analysis play a role in comparing gene sequences of pathogenic and non-pathogenic strains or fungi that attack crops, particularly chocolate?
Mm-hmm. Yeah, I think microarray could be used, but I think there's also other techniques that people could use, say an RNA-seq, which is a very, I think there's microarray, and then we had RNA-seq analysis, and over time that's become more streamlined, and sometimes can kind of be another tool that we would be able to use for that analysis.
And so that could help. I mean, if you determine pathogenic versus non-pathogenic, how would that be used in the field? I guess I'm not sure.
So I guess that people could. I mean, I understood the question to be like, how can you know which pathogens are or organisms are, or what makes them pathogenic? Is that that's kind of how I understood the question. Like, how can you? And the other thing, part of it is interesting, is that when you think about pathogens in plants, is that plants have these susceptibility genes. So they're genes that make them more susceptible to pathogens. So there's kind of a little bit of a flip side. Instead of trying to find plants that are more resilient by incorporating genes for resistance like the BT, can you identify these susceptibility genes and then knock those out and potentially use something like CRISPR or to knock those out and develop germplasm that breeders could use, that these plants aren't susceptible to these pathogens.
That makes sense. And throw a quick plug for next month's Breaking Ground. We'll be talking with Jim Giovannoni about ripening things, and specifically tomatoes, and keeping the skin tougher, and then things can't get in, diseases can't get in.
Oh yeah, right. Yeah, that kind of more of a physical barrier. Yep.
More of a physical barrier. Yeah, that's right. Shelf life. We have one from Lloyd. With food security initiatives expanding to include insect proteins as a primary source of food, how does this impact efforts in using plant biotech for the same end goal?
I think I see it as, my opinion is it's an option. It's another option for protein, for food supply. And I just don't think we should rely on one source or just a few types of sources. So I think if that's a viable option, that could be done. I'm not all that familiar with the insect proteins. I mean, I know about some, but I think I don't see them being mutually exclusive as far as what we're doing. I just think it's a good option, especially in regions where they might not have a diversity of sources for protein.
I love how BTI and PlantGENE are looking into succession planning and wanting to mentor young scientists. Will there be more outreach to raise awareness among students at all levels?
Definitely. Yeah, definitely. We talk about that. We talk about, trying to tap into the community to find out about options. I'm a big advocate of, get them young. We think about undergraduates, but I think we need to go to the other grade levels, like high school. I love our high school internship program here. And like I said, myself, as a high school student, I didn't know there was such a thing as plant breeding and genetics and crop improvement, and how it could really have an impact. Someone told me recently where, because I said, explaining how I wanted to be a doctor, and then I switched over, and they said, well, they heard us saying that as a doctor, you might help one person one-on-one, but in agriculture, if you're improving it, you help many. And I was like, oh, okay, that, to me is something that I think if we could kind of share that information with the younger generation and what options are out there. And it's not just that we're thinking about them going into it, but being supportive of it, kind of knowing where their food comes and how it's improved. So I just think one of the parts that excites me about PlantGENE is this training opportunity and looking at all different levels. And it's, having, like I said, the high school students having opportunities. We've talked about community colleges and looking again, as we all do, we want to have a diverse workforce and people from different backgrounds and to provide opportunities.
Yeah, community colleges could be great. And I know they often have like a lab technician program, just like a 2-year associates thing. There could be, try to get some plant transformation tacked in there somehow.
Yeah, that's the plan, is to kind of provide this, theory. I really want people to understand why they're doing what they're doing. I mean, that just helps all of us. And then also have this hands-on training. So we're really being strategic, especially with our advisory committee, to help us identify those opportunities at different universities and also within the community, getting that feedback. One of the things we're going to do in October is have a big launch event on October 4th, invite the community, and we have ways of tracking information about people want to see and what they're willing to volunteer to help with, and really start pulling people together.
That sounds great.
Get things done.
Another one from David Silberman. Several decades ago, we were told that transportation was a significant factor in getting food crops from stores to the populace. Is transportation still a factor?
I believe so. I think it's gotten better. And I think it's like people are thinking of also growing more locally, trying to find what they call these food deserts and trying to make it happen so that you don't have to worry about the transportation so much, is making it more accessible for people to have. And even transportation right now, we know it affects a lot of different factors of what we, food prices, food availability, and other, not even beyond food, but there are initiatives to make sure that we're getting food grown in particular areas.
Anyone else has any questions? Please put them in there in the chat box. We have one more comment right now. You could connect with the NSF program called Includes, that finds way to increase inclusiveness in STEM. I haven't heard of that one.
Oh, that's a great suggestion. Thank you. Yeah, no, I wasn't aware of that, but I will. I'm making a note right now to do that. Having these other, because we got the funding, but just knowing, are there other programs, and having people within the community identify these, having our coordinator try to help people identify opportunities. So that would be, that's a great suggestion.
I'm curious, I know this just launched in July, right?
Yeah.
A month old. But have you gotten a lot of bites so far? Like, what do you have on the website so far?
Yeah, so the website was the website's being hosted by actually a former collaborator who started his own business because he saw a need for these types of. Oh, excuse me. I'm losing my voice, but so on the website right now, it's a work in progress, but we have we recently, with the generosity of our collaborator and the Gates Foundation, who put funding towards these, they're called master classes. So there are these 34 classes, virtual classes, that are available. Myself and a number of them, I think almost all of, if not all, most of the steering committee participated in, and those are available. You can learn about different applications of gene editing, and then there's crop specific, like maize transformation. You can also identify people who are the leaders in those areas and get in touch with them. So we have that right now. We also have a join a kind of registration link where people can become a member of PlantGENE. And part of this is almost, I want to look at it like a dating site where you can, not that I have experience with that, but kind of this idea of where people could find those people who work in their areas and connect with them. Right now, unless you're reading publications or you know somebody who knows somebody, but just having these categorized, like, oh, I'm interested in cassava biotechnology. Well, here are the people working in cassava. Or I'm interested in this particular area of gene editing, and here are all those people that you can connect with. So we're building that membership, and we will have that platform available. And then we want to be able to have protocols up to be accessible for people to try. We want to have a helpline. We're we'll have these live sessions, or if people are having problems with whatever technique they're doing, is invite members of the community who can help. Also have a link where, if you're having a problem, this is the person to go to. These are the people, or these are the people in the area that can help you get through these stumbling blocks of what you're trying to do.
It's a great idea. It's kind of like, find a mentor, like phone a mentor.
Mm-hmm. Yeah. I always think of that Who Wants to Be a Millionaire or something, as like phone a friend kind of thing, or how to find a mentor. But definitely connecting people to try to help them, so they just don't give up. I mean, a big part of this, sometimes, people try something and it doesn't work and they're not sure why, and then it really can set their research back, and they might give up. But we want to be able to have people to find someone that could help them through those trouble spots.
Okay, I think that's all our questions for you, Joyce. That was fantastic. Thank
Great.
you so much
You.
for telling us about this really cool initiative that you're doing.
Yeah, thanks for the opportunity. I'm really happy to be able to share this with people, and like I said, I hope we can. Well, our plan is to have impact and get the community involved, and really bring people together to make this consortium type of to attack these issues.
And it sounds like it'll be a global.
Mm-hmm. Yeah.
Yep.
Yeah, definitely. I mean, it's very much, it has to be for us to have some sort of progress to be made. Yeah.
Well, thank you again, Joyce, and thank you everyone out there in Zoom land who joined us today. And please join us for BTI's next Breaking Ground discussion series on Tuesday, September 27th, with Dr. Jim Giovannoni, who will talk with us about his efforts to improve tomato flavor and shelf life. And you can register on our website. I'm about to, I just pasted a bunch of links in the chat box, and you can, it's going to be a really, really fun one. So please register at btiscience.org. And you can read more about BTI's current research and many other neat stories about BTI science in our annual report, which you can find online at btiscience.org/annual report. And BTI is an independent nonprofit research institute, and we operate in large part thanks to the generosity of community members like you. And if you'd like to make a gift to support BTI, you can donate online at btiscience.org/give, or email our development team at development@btiscience.org. And thank you, everyone. Thank you, Joyce. Thank you, everyone out there again for joining us. This was a great one. Lots of great questions. Lots of good engagement. These are so much fun. And I don't know. We'll see when we have you on for the third time, Joyce.
2 more years.
Yeah. All right. Thank you, everyone out there. Okay. Have a wonderful day and be well.